everyone, welcome to my YouTube channel BioLogics. I am Sahana Gaonkar. Let's learn together and let's start today's session. So today's topic is transposable elements. So these transposable elements are also known as jumping genes, mobile DNA elements, insertion sequences, etc. Coming to the next slide. So these moving elements are also known as transposons. So these transposable elements are short sequences of DNA that have ability to move from one location to the other in the genome and often make duplicate copies of themselves in this process. So the process by which the sequences are copied and inserted into a new site in a genome is called as transposition. So the first transposons were discovered in maize, that is Zea maize, by Barbara McClintock in 1948. So while she was studying the genetics of kernel mottling of the maize, she discovered an element that not only regulated the mottling but also caused the breakage of the chromosome carrying the gene for color and consistency of the kernel. So for this she awarded Nobel Prize in 1983 in Physiology and Medicine. Have a look. So the term transposable elements was coined by another greater geneticist, that is Alexander Brink, and based on the movement of these transposons they are classified into conservative process and replicative process. So the method of transposition is classified as conservative process and replicative process, that is cut and paste mechanism and copy and paste mechanism. So in replication transposition or replicative method, the transposon creates its own copy or the second copy of itself. So the original transposon remain in the place and the copy is inserted into a new position or to the target DNA. But in conservative transposition, the excision of the sequence in its original position takes place, which is followed by reinsertion of the sequence elsewhere in the target DNA. So in conservative transposition, it involves the change in the position of the transposon in the genome without increasing its copy number. Coming to the next slide, these are some general properties of the plant transposons. So plant transposon possesses ITR sequences and generate short repeats at the target sites. So what do you mean by ITR sequences? So these are the inverted terminal repeats that are structural features of the transposable elements that are essential for transposition process of certain DNA elements. So they can activate or repress the target genes, can cause chromosome mutations or disrupt the genes. So potentially we can say that they are the major source of genome diversity and change. Based on the mechanism of transposition, there are two types, that is DNA transposons and retrotransposons. That is, transposons are known as class 1 elements and these transposons or DNA transposons are known as class 2 elements. So both the transposons and the retrotransposons families have autonomous elements and non-autonomous elements. So this classification is done on the basis of their self-efficiency. So autonomous elements can transpose themselves and possess transposition gene. That means they are not dependent on any other helping transposon. So they are the independent element. They possess everything they need to promote their own transposition. But in non-autonomous elements, they do not transpose themselves. They lack transposition gene and rely on presence of another transposon. That means these elements depend on autonomous elements from the same family for the transposition. So we can say that they require a supporting gene. Coming to the next slide, retrotransposons or the class 1 elements, they are first transcribed into a RNA intermediate or RNA molecule. Then they are reverse transcribed into a DNA intermediate. After that they are integrated into a target DNA. But in DNA transposon, here the direct excision of the transposon takes place so that DNA intermediate is inserted into a target DNA. There is no need of transcription or reverse transcription. So we can say that these DNA transposons or the class 2 elements can move by cut and paste mechanism, and this retrotransposons can move by copy and paste mechanism. So these are some basic aspects of transposable elements. Let's study the other aspects of transposable elements in the next session. So if you like the video, please do support, share and subscribe. Thank you so much.